Hi friends, this is Naveen from AKN Sprint Education. Welcome to our YouTube channel. The topic that we are going to look today is the most important 8-mark question in biological science, right? The top most important question as I am expecting this year, explain any two natural and two artificial vegetative propagation methods to produce more number of plants in less time period with examples. Also they may ask question in different way. Explain the artificial methods of vegetative propagation in plants, or explain the method of artificial propagation in various plants. As children, vegetative propagation is a type of asexual reproduction that usually occurs in leaves, stems and roots. In plants, vegetative propagation is of two types. One is natural propagation, second one is artificial propagation. So natural propagation, in this type of vegetation, new plants emerge from the parent plant naturally. Leaves, small plants grow from the edges of the leaves. Example, bryophyllum. Stem, when stem like runner and stolon touch the ground, they produce the adventitious roots. If the connection from parent plant is broken, then the stem portion with adventitious roots will produce new independent plants. Such type of vegetation is observed in stems from stolons, bulbs, tubers, etc. Artificial propagation. Few methods of carrying out artificial propagation are as follows: cutting, layering, grafting. Cutting is a process in which a part of the plant is cut from the parent plant containing bud and grown individually to form a new plant. Example, rose, hibiscus. Layering is a process in which a weak branch from the plant is selected and viewed into the soil such that the tip of the branch is above the ground. After a few days, the part of the plant viewed under the soil will generate roots. Then this branch is detached from the parent plant and is grown independently. Example, Nerium, jasmine. Grafting is a process of obtaining one plant of desirable characters from two plants of closely related varieties. Example, mango, citrus, apple and rose. So children, this is the most important question. Coming to second question, which is also the most important question, heredity and evolution chapter. What is phenotype and genotype? Explain them with the help of Mendel's monohybrid cross. Children, as I am expecting this year this question, phenotype, genotype. The observable properties of an organism is known as phenotype. Genotype, the genetic makeup of individual referring a specific character is known as genotype. Monohybrid refers to experimenting with one character or trait. For example, consider pure breed of yellow Y seeds pea tree is crossbred breed with pure breed of green seeds pea tree. F1 generation. During F1 generation, when pure breeds of yellow seed plant is crossbreed with pure breeds of green seed plant, then all the plants obtained will be yellow only. Whereas in F2 generation, when these plants of F1 generation are self-pollinated, then 75% of the plants had yellow seeds and the remaining 25% has had green seed, right? So in the F1 generation plants, the plants produce yellow or Y, small y, capital Y, and the plants produce green. So children, what is phenotypic ratio here? Here phenotypic ratio is 3:1 and genotypic ratio is 3:1. So children, this is the most important question. You need to see here capital Y, small y, capital Y, small y, capital Y, small y, capital Y, small y, capital Y, small y. Here you see capital Y, capital Y, small y, capital Y, small y. So phenotypic ratio is 3:1 and genotypic ratio is 3:1. Okay children. Children, one more important question from chapter number 1. What process do you follow in laboratory to study the presence of starch in leaves? Else, explain the procedure and precautions to prove the presence of starch in leaves experiment. What is the aim of the experiment? To study the presence of starch in leaves, the following experiment is carried out. The materials required are checking the presence of starch in leaves when starch being a type of carbohydrate. So you can see here beaker, boiling water, leaf, methylated spirit, boiling tube, asbestos gas, tripod stand, Bunsen burner. So a soft and thin leaf well exposed to sunlight is taken from a plant. A test tube with water is taken and the leaf is boiled in it for few seconds. Later the test tube is filled with methylated spirit and the leaf is transferred. The test tube is now allowed to boil in water bath as shown in the figure below. As time passes, the leaf becomes pale as the chlorophyll present in the leaf is dissolved in spirit. The leaf is now taken out from the test tube carefully using brush. It is placed over a petri dish and few drops of iodine are added on it. You can observe the figure B. A bluish black color is observed on leaf and it indicates the presence of starch. What is the interference? From the experiment it is inferred that starch is produced during photosynthesis process. So what are the precautions you must have taken? Make sure the level is completely inserted into the test tube with methylated spirit. Carefully transfer the leaf from the test tube to the petri dish using the brush. Children, as I am expecting this year this question only, so read carefully. Coming to one more important question, list out the materials required and the procedure to be followed to prove that carbon dioxide is essential for photosynthesis. So what is the aim? To check whether carbon dioxide is essential in the process of photosynthesis. Requirements: a detached plant, that is a healthy plant, is stored in dark place for at least 3 days. By this, the starch present in the plant leaves is removed, thus making the plant a detached plant. Potassium hydroxide KOH pellets are solution. It absorbs carbon dioxide. A wide-mouthed transparent bottle and a spirit cup. Procedure: a wide-mouthed transparent bottle is taken and is filled with KOH pellets as solution. The opening of bottle is fixed with a split cork and one of the detached plant leaves is inserted into it as shown figure below. Now the whole setup is placed in sunlight and left undisturbed. Few hours later, the leaf inserted in the bottle and any other leaf of the plant are taken. They are tested for starch presence. That is, place the leaf in petri dish and add few drops of iodine on it. After testing the leaves, it was observed that the part of the leaf in the bottle remained colorless and the part of leaf exposed to the atmospheric air and the sunlight turned to bluish black, indicating the presence of starch. So what can we conclude? As the KOH solution present in the bottle absorbs the carbon dioxide, the detached plant leaf did not regain starch. This clearly proves that carbon dioxide is essential for plants to undergo the photosynthesis process. Children, diagram is also important. Coming to one more important question from chapter number 3, transportation. What is called pumping station in human body? Explain the structure with suitable diagram. Sometimes they may also ask internal structure of human heart. Heart is called pumping station in human body. Heart is called pumping station in human body. Heart is pear shaped and its outline is triangle shaped. It is covered by two-layer membrane which are separated by a fluid known as pericardial fluid. Internally, heart is divided into 4 chambers. Upper two chambers are called arteria or auricles. Lower two chambers are called ventricles. The walls of the ventricles are thicker to atrial valves. These chambers are separated by septa and connected through valves. 5 major blood vessels originate from the heart. They are superior vena cava, inferior vena cava, aorta, pulmonary artery, pulmonary vein. Superior vena cava collects blood from upper parts of the body. Inferior vena cava collects blood from lower parts of the body. Aorta supplies blood to all body parts. Pulmonary artery carries blood from heart to lungs. Pulmonary vein carries blood from lungs to heart. Tricuspid valve is the valve which connects a right auricle and right ventricle, and bicuspid valve is the valve which connects left auricle and left ventricle. Coronary arteries supply blood to the muscles of the heart, and coronary veins are veins which collects blood from the heart. Children, even you need to draw the structure of the human heart and you need to label the parts. So, drawing a structure of human heart is not a difficult one. Once you go through YouTube short, so they will show you. And one more important question. Explain the procedure and observation of the experiment conducted to observe the internal structure of kidney. To observe both internal and external features of a kidney, the materials required are goats, specimen of goat or sheep, kidney, a blade, tray and water. Wash a kidney with water thoroughly until the blood is completely removed. Keep it in the tray and study it. Make a longitudinal section with the help of scalpel. Study the internal structure and write down the observations. You need to observe the diagram as well as external view and internal view, and try to observe the parts. If you observe that kidney which is in bean shaped, the color of kidney is reddish brown. A cap like structure called adrenal gland is seen on upper portion of kidney. In longitudinal section of kidney, dark brown color portion is found on outer zone of the kidney. 2 tubes are coming out of the kidney future. You can see that urethra, renal vein, renal artery. This is renal vein, this is renal artery. You can see that interlobar blood vessels, cortical blood vessels, minor calvix, major calvix. See capsule, and this is medulla. Okay children, so internal structure of kidney, observe the experiment conduction, right, which is also the most important question. Now coming to one more important question. Explain the temporary and permanent methods to be adapted for kidney failure. Kidney failure. For kidney failure person, dialysis is a temporary solution and kidney transplantation is a permanent solution. What is temporary solution? Dialysis. What is permanent solution? Transplantation. Dialysis. The impure blood is taken out from the main artery and mixed it with the anticoagulant such as heparin. This blood is now pumped to the dialyzer. In dialyzer, blood moves through the tubes made of cellophane. These tubes are separated by the dialysing fluid. This membrane will filter the waste products from the blood and moves it to the dialyzer fluid through the diffusion process. Then this filtered blood is pumped back to the body through the vein after adding anticoagulant. It takes approximately 3 to 6 hours to complete dialysis session. This process is used all over the world for thousands of uremic kidney failure patients. Then permanent solution is kidney transplantation is the best long-term solution for kidney failure. For transplantation, the donor must be close relative who can donate a functioning kidney. The immune system of the recipient usually tries to reject the transplanted kidney. To avoid this, the kidney has to be good match to the recipient body. However, success rate of such complicated techniques is increasing gradually due to modern clinical procedures. Nowadays, a lot of kidney failure patients can be saved by organ donation. Organs are collected and then transplanted from brain dead patients to the recipient. Okay, children? Coming to next question, what procedure do you follow to understand anaerobic respiration in your school laboratory? Or explain the experiment to understand the respiration process in yeast. Or else write the procedure and observations of the experiment which you have conducted in your laboratory to prove that CO2 and heat are evolved during anaerobic respiration by using yeast. So children, the aim of an experiment to understand the anaerobic respiration in yeast, materials required: thermoplast, wash bottle, thermometer, glass tube, glucose solution, yeast cells, liquid paraffin bicarbonate solution. Procedure: take a glucose solution in thermos flask and heat it for a minute and then cool it without shaking so that the dissolved oxygen from the glucose solution is removed. Now add yeast to it and add few drops of diazine green. So that is called Janus green B solution. Then add 1 cm layer of liquid paraffin to stop the oxygen supply to the mixture. After some time, the color of mixture changes to pink, which indicates the oxygen content is reduced from the mixture. Now insert thermometer and another glass tube such that the another of glass tube is connected to the wash bottle as shown below. This arrangement is made such that gas escape from the thermos flask and enter into the wash bottle with lime water. To speed up the reaction, apparatus can be heated up to 37 degrees centigrade. If the arrangement is kept undisturbed for 1 or 2 days, gas is evolved from the thermos flask and turn lime water in the wash bottle to milky white precipitate. What can we conclude? Yeast cell respires anaerobically, that is in the absence of oxygen, and converts glucose to ethyl alcohol, releasing CO2 and heat energy. So children, these are the most important gunshot 8-mark question from biological science. So children, don't forget to hit subscribe button, like it and share it with your friends. Thank you children.